So it was inevitable. We had to eventually talk about antimicrobial, more specifically antibiotic resistance. It's the sad reality of today in healthcare, and it's not going away anytime soon. Drug resistance is formally defined as an adaptive response in which the microorganisms begin to tolerate any amount, maybe a small concentration or a larger concentration of the drug that would have normally been inhibitory or killed off that microorganism. Drug resistance has been on the rise. It's been exponentially increasing as the genes that code for drug resistance have been put in plasmids by the microorganisms in nature themselves, and then those plasmids conferring resistance are being transferred from microbial species to microbial species via conjugation. This genetic versatility and adaptability of the microbial populations is tough. It's inevitable. It's going to happen. It's an excellent example of microevolution, and it's been very well documented. It can be intrinsic with the microorganism. Sometimes the genes that confer resistance are going to be in that large nucleoid region with a large single chromosome of the microorganism, for the bacterium at least, and sometimes the genes that confer resistance can be transmitted from organism to organism via a plasmid. So, penicillin, that first antibiotic, that glorious first antibiotic, almost immediately had resistance developed to it. Recall from a couple earlier in this chapter, I had mentioned that the first kind of penicillin was very limited in its scope because it did not have a very broad spectrum of action. That very first penicillin was very targeted, very focused, and resistance to it developed almost immediately. So that was when the first antimicrobial resistance or antibiotic resistance was known, but back in the 40s, 50s, and 60s, it was a golden era of antibiotics because new antibiotics were constantly being discovered. Then, in the 1980s and 1990s, scientists began to notice large-scale treatment failures of antibiotics. This also corresponded to the halting of antibiotic discovery. Very few antibiotics have been discovered since the 1980s. I read an article about one that was discovered in late fall of 2014, and I was super duper excited about that because for my own well-being and the well-being of my family and my country and society, I really hope that we can have some more antibiotics in the pipeline right now, so to speak. Microbes that can become resistant to a drug if one of the following things occurs. We could have a spontaneous mutation in a chromosomal gene, or we can have horizontal transfer. So we can have a sex pilus transmit that plasmid, or we can have the gene be admitted into the outside environment, so we can have transduction take place, or we can have a bacteriophage transfer the gene from one bacterium to another bacterium. So let's talk about chromosomal drug resistance. This is usually going to be from a spontaneous mutation that's non-directed and random, and it's going to cause a slight change in drug sensitivity. It's not going to be incredibly dramatic. So we have chromosomal-based drug resistance that can typically be overcome by increasing the dosage of the medication. The persisters are going to be the slowly or stopping of the metabolism in the microbe that can't be harmed by the antibiotic. So in other words, these persisters are microorganisms that can put their metabolism on pause when they're exposed to the antibiotic. And then they wait for the antibiotic to be processed or diffused out. And then once that antibiotic leaves their cell, their cytoplasm, they restart their cytoplasm and then continue with their nasty microbial effects on the human host. Resistance through horizontal transfer is the one that, generally speaking, is going to be a little bit scarier. There are going to be resistance factors, or R factors. These are plasmids, little circular pieces of DNA that are going to have a specific gene or genes that confer antibiotic resistance. They can be transmitted through conjugation, transformation, or transduction. Remember, conjugation is direct linkage between the cells, cell-to-cell linkage. So that's the sex pilus linkage. Transformation is when the naked piece of DNA is excreted into the extracellular environment and then uptaken by a random cell that comes in contact with it. And then transduction is when a bacteriophage is going to transmit the gene from one cell to another cell. Good coffee. So, if we look at the plasmids that code for drug resistance, these plasmids are naturally present in microbes all the time, even before they are exposed to the antibiotic. The thing is, these plasmids were present in very low concentrations in the microbe microbial species. But once we started bombarding the microbial populations with antibiotics, we added a new selective pressure so that the microbes that had these plasmids had a reproductive advantage and became a much larger part of the population compared to the microbes that did not have those plasmids. We could also have transposons. Remember, a transposon is that jumping gene, a gene that can duplicate itself and move around within the genome of the microorganism. Transposons can duplicate and insert drug resistance genes from a plasmid to a chromosome, from the chromosome to the plasmid, and generally propagate those resistance genes. The fact that one microbial species can share the same resistance gene with a separate microbial species has contributed dramatically to the proliferation of drug-resistant species. So, I love this figure. We're going to spend some time camping on this figure. I love this figure because it goes into just the right amount of detail. It doesn't bombard you with lots of nitty-gritty little details that you won't be expected to know unless you're a physician or a pharmacist. This is a good overview of antibiotic or drug resistance. So one mechanism for antibiotic resistance is the microbe develops an enzyme that deactivates the drug. So let's say we look at our generic penicillin molecule with its generic R side chain, and the penicillinase is an enzyme that breaks down penicillin. It's going to add a new functional chain over here. So this is a carboxylic acid functional group over here. It adds a new functional group to the side chain and deactivates or removes one of the rings from penicillin. So this penicillinase is going to digest or deactivate penicillin. So mechanism number one, the microbe develops a new enzyme that destroys the drug molecule. Mechanism number two, the microbe can no longer absorb the drug molecule. Maybe before the mutation, the drug could bind to a surface receptor and make it into the microbe, no problem. So we had normal surface receptors. But maybe those surface receptors on the microbe were mutated now. Now they have a different shape, and the drug can no longer go into the microbe. It just boing, bounces out the surface of the microbe. So we could maybe have a mutation that makes the drug cannot be absorbed by the microbe anymore. What's mechanism number two for drug resistance? Mechanism number two is that the drug can still be absorbed by the microbe, but now the microbe has a pump that can pump the drug immediately out of its cytoplasm. So the drug is absorbed, but immediately the drug is ejected from the cell. So there can be a mutation that occurs that will code for a cell transporter pump that will pump the drug out of the cytoplasm of the microbe before the drug can cause damage. Another option is the binding site of the target, the binding site that the drug target could change. A common target for antimicrobial treatment, in particular antibiotic treatment, is the bacterial ribosome. And here we can see a triangular-shaped drug molecule, because of course drug molecules look like green triangles, and it's binding to the large subunit of the ribosome. There's my mouse. All right, there's my mouse. Okay. But what if that binding site goes from triangular to circular shape? If that happens, the triangular drug molecule can no longer bind to the large subunit of the ribosome, and the drug is rendered ineffective. So we could have, there we go. So we could have the binding site for the drug change shape. And then the final option, the fifth way that we can have antimicrobial resistance occur, is the microbe can develop a brand new metabolic pathway. Let's say the drug blocks the conversion from molecule A to molecule C. And that blocking of the metabolic pathway keeps the microbe from making its necessary end product to stay alive. What if, though, the microbe developed an alternative metabolic pathway that still gave it the same end product? In that case, the microbe could work around the fact that it's being treated by an antimicrobial, and the microbe would be resistant to that antimicrobial drug. So, to summarize our 5 different methods, there can be a mutation. When we look at natural selection and drug resistance, as drug resistance develops, there are going to be very long-term therapeutic consequences. Any population of microbe is going to almost always have a few cells that have microbial resistance. And I should say any large population of microbes. And since microbes are so small and they reproduce so quickly, it's pretty easy to get a large population of microbes. Now, in those large populations of microbes, if the drug is not present, the gene typically is not expressed for the microbe. So early on, if you have that first, if you look at a virgin population in a patient, so let's say you have a patient come to you, they're sick with a bacterium, and that patient has not been taking any antibiotics, even if those bacterium in the patient have genes that code for drug resistance, those bacterium are probably not actively expressing those genes. Remember when we talked about the lac operon? Microbes can turn genes on and off depending on specific chemicals they are exposed to. So if the microbes have not been exposed to the antibiotic, they may not be expressing the gene, or they may not have turned on the gene that makes them resistant to the drug. However, if we expose that population to the drug, then the drug-resistant population can turn on the genes that they have to make them drug-resistant, and now they're going to have some kind of selective or competitive advantage. They are going to be able to reproduce quicker and better than the microbes that do not have the drug-resistant genes. And the offspring will inherit drug resistance, and the new population, after several generations, is going to have a large percentage of them with the drug-resistant form of the gene. So now most of the microbes in that population are going to be resistant to the drug. So let's say we just have a tube of a nutrient broth full of drugs, or full of microbes, and we plate it. Most of the microbes in that nutrient broth probably are not going to be resistant to bacteria. And if I plate those microbes right away on a nutrient plate that has antibiotics in it, most of the microbes are going to die, but I'll get I'll get a few colonies. However, if I put some antibiotics in my test tube and then wait several hours so I can have several generations of bacterium reproducing, and then I plate my bacteria on a nutrient agar plate that contains antibiotics, I'll have a lot more growth because there's a much higher percentage of the population that is actively resistant to the antibiotic. This is a big problem. In 2013, a threat report issued by the Center for Disease Control outlined some very catastrophic, and I'm just going to call it there and say totally catastrophic, not potentially catastrophic, totally catastrophic scenarios related to antibiotic resistance in general populations of microbes. This is the kind of stuff that keeps me awake at night. The stories or the scenarios that you hear about where you can't have organ transplants anymore or open surgery anymore because you need to have an antibiotic regimen during open surgery, or you can't treat common diseases that we are used to treating now, and now it would take us back to the Middle Ages in many respects for drug treatment. It horrifies me. New and effective antibiotics are very slow to come to the market. Antibiotics are not economically lucrative. You do not make money by making an antibiotic because antibiotics cure an illness. And making an antibiotic is an incredible investment of resources. Drug companies also do not make money by making vaccines. Drug companies will make money by coming up with a therapy that treats the symptoms of a chronic condition. What do I mean? I mean something like a high blood pressure pill, a high cholesterol pill, some kind of a pill that you're going to have to take every day for the rest of your life. That's how a drug company makes money. Antibiotics and vaccines are very low profit yields. There's very, very little money in it for the drug companies. So because of that very low amount of money that for the drug companies, there's very little financial incentive for the drug companies to develop these. Pfizer was the last private drug company in the United States to have an antibiotic research team. And as of right now, there is no drug company in the United States that is actively researching new antibiotics. All research that's being done for new antibiotics is being done in academia or in university settings. Something else should be mentioned is that it takes years and years and years to make a new antibiotic come to market. It takes at least a decade. And can you imagine paying a laboratory team
or a team of people for over a decade without getting any results that you can take to market to make any profits from. So you need to have very deep pockets to fund this team of very intelligent, very expensive people for a decade or more before you can get a return on your investment if you get a return on your investment. So from a purely capitalistic perspective, there's not a lot of incentive to make new antibiotics. So, what are some antibiotics right now that are urgent threats? We can have Clostridium difficile, and then Carbapenem-resistant Enterobacteriaceae, and drug-resistant Neisseria gonorrhoeae. So these are rapidly spreading through the developed world right now. We have some more serious threats as well, and there are numerous. This is legion right now, serious threats. We have multi-drug-resistant Acinetobacter, drug-resistant Campylobacter, fluconazole-resistant Candida, and many others. And there are also some concerning threats, vancomycin-resistant Staphylococcus aureus. Dang, if the Staphylococcus aureus is resistant to vancomycin, we have a problem because vancomycin has been our last reserve or our last holdout. It's the newest antibiotic that hit the market. It's the youngest antibiotic to hit the market, so that most microorganisms are still susceptible to vancomycin. And now we're already having vancomycin resistance hit the populations. Erythromycin-resistant group A Streptococcus and then clindamycin-resistant group B Streptococcus are also concerning threats. I also want to throw out there multi-drug-resistant tuberculosis. I spend a fair amount of time reading about, especially while I'm teaching the course, reading about microbes and hitting journal articles. And here we have a journal article from our World Health Organization about multi-drug resistant tuberculosis. Another term for this is pan-resistance. Pan is a root word that means the world. Think Pangea, for example. And if you have a pan-resistant microbe, it's resistant to every treatment that's present in the world. Pan-resistant tuberculosis right now is spreading through Southeast Asia, it's spreading through the Russian prison system, and it's spreading through Africa. And it is a plane ride away from hitting North America. Pan-resistant tuberculosis has no known treatment option and no known cure. The only treatment option for pan-resistant tuberculosis is hope to God and pray that your immune system fights it off. Back to the PowerPoint. Boom, there we go. So, we need to come up with a new approach for antimicrobial therapy. Antibiotics are becoming a thing of the past. They are not financially lucrative or financially viable to produce new antibiotics. And with in an era of government budget cuts, cut here, cut there, balance the budget, you are not very likely to find an increase in government funding for antibiotic research. So let's target something other than, or let's come up with a new target. Many bacteria need iron, for example, to reproduce. And iron is a limiting nutrient for many bacterium. So if we can target the abilities of a bacterium to scavenge iron molecules from the host, we could potentially limit growth that way. So we could target, reduce the amount of iron present in the host. We could chelate those iron molecules, maybe using disodium EDTA or sodium citrate as some chelating molecules that I came up with off the top of my head. Or we can focus on RNA interference. RNA interference is a new kid on the block. We can focus on destroying RNA molecules as they're being expressed. And if we have a messenger RNA molecule that we destroy before it is translated, we can never get the protein that the messenger RNA molecule was coding for. So if we can eliminate the messenger RNAs, we will shut down the metabolism of the cell that no longer has that particular messenger RNA present. Messenger RNA interference strategies have been found to be effective for both bacterial and particularly viral infections. We could also mimic defense peptide molecules. When we get to the immune system chapters, because we have multiple chapters on the immune system, we will talk about some small categories of molecules that are present. They're larger than an antibiotic. These are polypeptides, so they're going to be between 20 and 50 amino acids. These small
proteins and relatively large molecules, when you compare them to the size of an antibiotic, are just secreted as part of the innate immune system. And we'll talk a lot about the innate immune system in a chapter or two. So if we can mimic the innate immune system of other organisms, or ramp up or supercharge our innate immune system, we are going to be able to fight off more infections. One thing that's worth emphasizing is a lot of research is being done right now on crocodile and alligator innate immune systems. These are, large organisms that live in swamps and cesspools and have cuts and lacerations all the time. But you never ever see them get an infection because they have such an excellent innate immune system. They have defense peptides that are very well established and fight off a plethora of infections. Bacteria also produce defense peptides, and if we can mimic these defense peptides, we can produce them and insert and give them to a patient to fight off potentially bacterial infections. These defense peptides, generally speaking, are going to insert into the membrane of the bacterium or potentially other targets in the cells, and they can be more effective than narrowly targeted antibiotics because they have very broad actions and they're going to be more difficult to develop resistance to, hopefully. One that I'm particularly excited about is bacteriophage therapy. So instead of using a drug to destroy a bacteria, why not use a virus to kill the bacteria? Have a specific virus, a bacteriophage, injected into the patient that destroys the bacteria that's causing the infection. Eastern European countries have been experimenting with bacteriophage infection for years now to complement antibiotics. These bacteriophage-based therapies have never been approved for use in Western countries. Biophage PA has been used to treat ear infections caused by Pseudomonas aeruginosus. Biofilms, that's exciting. These bacteriophage therapies are able to penetrate biofilms. Other research has been done to incorporate bacteriophages into wound dressings. And then finally, if we look at our bacteriophages, the big advantage of them is their narrow specificity. Bacteriophages, generally speaking, will only infect one species of bacterium at a time. So a big advantage of bacteriophage therapy is that you can destroy, potentially, the pathogenic bacterium, but leave your normal and large bacterial biota of your large intestine intact. For those of you that have worked in a nursing home and have experienced the joy of a C. diff infection induced by a strong antibiotic regimen, you know exactly what I'm talking about. When a patient takes a strong antibiotic regimen to kill off an infection, generally speaking, they kill off lots of the bacteria in their colon, and they're at risk for having Clostridium difficile overpopulate in their colon and cause a secondary infection. We can help nature along. Probiotics are microorganisms that are fed to us, that are beneficial for us. Probiotics are commonly prescribed after strong bouts of antibiotic therapy or antimicrobial therapy, and they can augment the normal bacterial biota that are already present. So if, for example, your kid is taking some antibiotics, you can do your kid a favor by getting some yogurt and just mixing some yogurt in with the food and giving your child some extra yogurt while they're on their antibiotic therapy and immediately after they're done with their antibiotic therapy to help keep their intestine populated with healthy commensalistic bacterium. Probiotics are very safe. There are almost no known side effects. One side effect could be you could gain some weight because you eat way too much delicious tasting yogurt. Probiotics are very effective, and it's been found to be useful in the management of food allergies. Prebiotics, unlike probiotics, a prebiotic is a nutrient that encourages the growth of the bacterium. So these are going to be supplying limiting nutrients to maximize the growth of beneficial bacterium. A fructan has been found to encourage the growth of bifidobacterium in the large intestine of human patients and discourage the growth of other pathogenic bacteria within the large intestine of human patients. Prebiotics are not as well understood as probiotics. When in doubt about probiotics, if you buy any kind of food at the grocery store that has live bacterial cultures in it, there is a very, very strong chance that those live bacterial cultures are probiotic, even if the label doesn't necessarily advertise probiotic on it. If it's the food contains live bacterial cultures, it's probably going to be at least mildly probiotic. And one that I find particularly interesting is a fecal transplant. These can be used to treat recurrent Clostridium difficile or C. diff infections or ulcerative colitis. What do? We give the sick patient, the patient that has the problems, we give them laxatives to clean out, flush out their colon, and clean as much of the poop or feces out of their large intestine as possible. And then we take feces from from the large intestine of a healthy patient and insert it through the back door, so to speak, into the patient that was sick, so that we can give that patient good, healthy bacteria in their colon to help hopefully relieve the effects of the large intestinal infection. Fecal transplants to treat C. diff infections have been found to be over 80% effective, dramatically more effective than the traditional route of giving the patient more antibiotics, which ironically triggered the C. diff infection in most cases. So, class, concept check. Which of the following mechanisms of antibiotic resistance act specifically on penicillins and cephalosporins? So, to answer this question, you first need to think, what do penicillins do and what do cephalosporins do? Our penicillins are going to focus on producing or decreasing cell wall synthesis. You'll need to slide back in your PowerPoints to find the answer to this question. Our options are A, decreased permeability or uptake of the drug. B, our enzymes are synthesized, inactivating the drug. C, binding sites for drugs are decreased. D, alternative metabolic pathways are used. Or D, drug is immediately eliminated through a transport pump. If you don't know the answer, that's okay. You can look it up in your textbook. You can flip back in the PowerPoint, or you can rewind the video. So pause the video and get me an answer. 5, 4, 3, 2, 1. So you may remember from a table that I had earlier in this presentation. Here we go. I talked about how a new enzyme can develop that could deactivate the drug, and I highlighted how we can deactivate penicillin. And if we read in the example here, that both penicillins and cephalosporins can be deactivated by the production of new enzymes, penicillinases. So, the answer to our question is B, enzymes are synthesized, inactivating the drug. If you have any questions about this or other topics that we talked about in this video, please feel free to post them on the discussion board or email me. Happy studies, class!